Bom dia a todos, bem-vindos a este Encontro Ciência Online, neste novo formato. And now switching to English, since our session today will take place in English. So welcome everybody, thank you for joining us in our new format, in our Ciencia meetings. This is the first of our Ciencia meetings online. And we are very happy to have today Ana Rita Carlos, our researcher. Today we also have Professor Solveig and Professor Cristina Magos, to whom I will give the floor very shortly. So my job here today is just to welcome you and just to let you know that during the talk, if you have any questions, please leave them in the live chat that you have on your right. If you have to log in with a Gmail account, but please feel free to leave your questions on the live chat that you have on your right. We will give them to Ana Rita Carlos by the end of the talk, so please feel free to leave your questions. And now I will give the floor to our coordinator, to the C3C coordinator, Professor Cristina Magos. Thank you.
Muito bom dia, ainda é bom dia. So, okay, in English. So I'm just here because this is a historical moment that we are experimenting. So the good part of this pandemic COVID-19 is the fact that we need, all of us, we need to adapt to the new technologies. And of course, C3C is in the front of this adaptation, and I really would like to welcome all of you, and I will encourage you to participate actively on our online talks. Of course, that today it's a very important day because we are going to have Rita Carlos that Solveig will present. But from now on, be aware that our Ciencia meetings are coming back, and it's our pleasure. And I also would like to thank, in particular, to Martin, to Mariana, that is helping a lot in the establishment of all the technical conditions. And from now on, we will be always online, and our Ciencia meetings will be successful as before. So thank you very much to all and welcome.
Bom dia a todos. It's a pleasure for me also to be here in the first Ciencia talk. And it's a pleasure to introduce Rita Carlos, who has been with us in C3C for a year now. She's originally from Faculdade de Ciências. She did her undergraduate degree in biology and went on to do the master's in molecular biology and genetics, where she worked in the lab of Rogério Tenreiro with Teresa Semedo doing her master's thesis. Then she went to the UK, to the University of Oxford, and worked with Madalena Tersones on telomere dysfunction and genomic instability in cancer. And in 2013, she came back to Portugal, to the IGC, where she worked in the lab of Miguel Soares on disease tolerance and cellular stress against disease. And she has joined our group a year ago, like I said, and she is going to talk about how she's using her expertise in helping us to understand muscle disease. And this is going to be her subject in this talk. So go ahead.
Good morning, everybody. Thank you for the kind introduction. So I'm just now going to share my screen with everybody. Okay. So as Solveig just mentioned, I'm going to talk about mechanisms of muscular dystrophy, muscular disease, in particular the disease caused by mutations in the LAMA2 gene, which leads to a congenital muscular dystrophy. So to start, just to give you some feedback and some background, congenital disorders, as defined by World Health Organization, are structural or functional anomalies that occur still in utero, or that can be identified only at birth. And in some cases, they may be detected later in infancy, as for example, hearing disorders, which are quite difficult to identify before at birth time. So these disorders, they can have different sources. They can have genetic impact, can have environmental, they can be promoted by infections, or also linked to maternal nutrition. So in terms of genetic, they're all inherited mutations, usually are recessive mutations, and they are really promoted by this fact by inbreeding. So that's why in certain communities, some congenital abnormalities are well established because of this inbreeding. And then we look at the environmental part, and the environmental, it can be seen just as a small part of the environment, or environment can actually take over everything else that is not genetic. So here we are going to assume that environmental will be everything. So infections and maternal nutrition is also part of our own environment. And they can have, for example, linked to teratogenic agents, so agents that will produce defects during embryonic development. And if we think about what is environmental risk, we can see either a personal risk. For example, if the mother during pregnancy takes certain drugs or consumes certain substances that can be toxic for herself and then for the baby as well. But there are also an environmental risk caused, for example, by people that live in nearby certain areas, for example, close to mines that can be very toxic environments. Now, moving on to muscular diseases, and in particularly, we are going to talk about muscular dystrophies. This is actually a very broad group of diseases, which all have in common the progressive skeletal muscle wasting and degeneration. So here, just a scheme of these diseases. And most of the diseases, actually, they are either part of extracellular matrix, so protein, different proteins that aligned outside the cell, and they give very, very important structural and physiological support to the cell. So it's proteins and other molecules that allow this the cell and the tissue to be stable. So they can either be mutations on this extracellular matrix, or receptors or linkers between this extracellular matrix and the cell cytoskeleton. And here you can see, for example, so the protein that I'm going to talk about today, which is this LAMA2. So it's causing mutations in laminin alpha-2. It's part of an extracellular matrix, but you have, for example, mutations here in another protein which links extracellular matrix with cell cytoskeleton. That is the cause of Duchenne muscular dystrophy, a very well-known muscular dystrophy. Okay, so considering the importance of the skeletal muscle to give support to the body, it's not very difficult for us to understand that actually people that suffer from these conditions have their life quality severely impaired and very reduced life expectancy. And I'm now talking about, in general, muscular dystrophies, but in particularly what distinguishes then what is a congenital muscular dystrophy from the remaining ones is this fact that I mentioned in the very beginning. It's that it's present right before birth or at birth. These congenital muscular dystrophies, or CMDs, are mainly caused by genetic mutations, but environmental factors do play a very important role, and in particular the infections. And then I'm going to come back to this when I start talking now about LAMA2-CMD in particular. So LAMA2-CMD, as I mentioned to you, is a congenital muscular dystrophy caused by mutations in LAMA2, which then codes for this laminin alpha-2 chain, so component of the extracellular matrix. So here you have again depicted how laminin, so alpha-2 will be this chain here, which then has specific receptors in the cell that then links to the cell cytoskeleton. So patients that suffer from this severe neuromuscular disorder have hypotonia, so they have no tonus in the muscles, they have muscle weakness, and often, because this affects the muscles, all the skeletal muscles of the body, they have inability to breathe and swallow. The prevalence of this disease usually is defined as between 1 in 30 to 1 in 400,000. It varies as well according to the data that we have available for different countries. Most patients, unfortunately, fail to survive past adolescence, except for a very small group of patients that suffer from a milder form of the disease, which is associated with a late onset. As I mentioned to you, the environment plays a critical role for the severity of this disease, in particular infections. And this occurs as the muscles are not, so they're very weak. The respiratory muscles are all very affected. So these people have very difficulties even to inhale and exhale, and also to cough. So this leads to accumulation and rotation of secretion that promotes then the growth of different microorganisms and potentiates respiratory tract infections. And that's why it comes as the cause number 1 for lethality in this disease. But so I told you a bit about what is the disease and the symptoms, but what are the mechanisms that are associated with LAMA2-CMD? So I made this graph to try to understand what we know and what is unknown. So if we think about what is our knowledge and how the disease progresses, so we have the onset of the disease, which is still not very well established for humans, but we believe that is before birth. Then we will have at birth, and then we have the first weeks of development of the child after birth, and then we have very clear symptoms that develop and that are promoted as well by secondary events, which are primarily chronic inflammation and fibrosis. And if we look at the graph, what we know the most actually is about the secondary events. So events later on in the development of the disease, and which are the ones that in terms of clinic are targeted. So this is a disease that has no cure so far, and the treatments are made to ameliorate the life quality of the patients. And then we have this intermediate pinkish area here, which is after birth in the beginning. So the very first symptoms development, and people are studying what is happening here. So using primarily mouse models of disease. And then we have the onset. So the very first moments when do we require in wild type in situation, when do we require LAMA2 to be present, when starts it to be so critical that the disease starts developing. And this is actually what we are interested in the development and evolutionary morphogenesis lab. So as I told you, what is known? So what is most known is the mechanisms for this secondary events, so chronic inflammation and fibrosis. Just a brief overview. So in a normal, healthy individually, when we have an acute injury, it's normal to have immune cells that come into the tissue to allow the repair of this tissue, and then they leave the tissue after the regeneration is completed. In a chronic injury, what we have this infiltration of immune cells, but they stay there and they become much more cells. And they change as well the production of various molecules that in the end leave a fibrotic scar. So here in this diagram on the right, we see that primarily we have as immune cells that act macrophages. So we have both pro-inflammatory macrophages that will promote the first steps, and then anti-inflammatory macrophages that will promote the tissue regeneration. So in a normal, healthy individually, in the case of acute injury, we have the cells that produce profibrotic factors as TGF-beta, for example, and then this stimulates, for example, fibroblasts to produce extracellular matrix. And this is important because it remodels the environment and allows the repair. In the case of chronic inflammation, what is happening is that the cells are always there. So immune cells are always there. Macrophages are much more, so there's more infiltration. There's more production of the signaling molecules. And for that reason, there is a really strong profibrotic program being activated. There is an excess of extracellular matrix, and there is this fibrosis that is very bad for the tissue. And this accounts for the pathogenesis as well of LAMA2-CMD. And this is what is actually being most treated in terms of the patients, as I mentioned to you before. So in this representation here, we have different molecules, signaling molecules, extracellular signaling molecules, and also receptors, and then effector molecules that will activate this fibrotic transcriptional program. And in this pinkish color, you can see various inhibitors that some of them that are already currently being used in terms of clinics that target this signaling pathways and that try to abrogate the fibrosis transcriptional program. So, but what other mechanisms apart from fibrosis and chronic inflammation? So there's another mechanism that is also very well studied, not so much in terms of therapeutic point of view, but in terms of research. Various studies performed in mice and using human cells as well have also shown that apoptosis plays a very important role for the pathogenesis of LAMA2-CMD. So apoptosis is a cell death pathway, which will mean that the cells, the muscle cells, will undergo cell death. So in these patients, so there is an increased number of cell death in LAMA2-CMD patients. So in this study that I'm showing here, what the authors did was actually they used a LAMA2-deficient mouse, which they crossed with a Bax mouse. And Bax is a protein, is a apoptotic protein. So it's a central player in apoptosis. So if we use a double knockout, actually, so in a single Bax knockout, there is no apoptosis, so the apoptosis pathway is impaired. And when we crossed LAMA2 with the Bax, we are inhibiting apoptosis in the LAMA2 background. And when the authors did that, they actually revert the survival of LAMA2 knockout mice, which is the red line here in the graph in figure A, which was about up to 70 days, and they were able to revert. So 100% of the mice survived during the time that the 120 days that they monitored the mice using the double knockouts. And they also improved strongly in terms of their body weight and body mass. So here you have on the LAMA2 wild type mouse, which is then similar to the double knockout LAMA2 Bax knockout. And you can see as well here the single knockout for LAMA2 mutations, which is much smaller than the remain ones, and the muscle mass is much smaller as well. So this just to show you how important it is apoptosis as well in the context of LAMA2-CMD. But there's still much to know. So the apoptosis, apoptosis is also a consequence of something, right? So people are still wondering, so what is happening before that? So different approaches have been taken, and people are also looking at either proteomic or transcriptomic approaches to try to understand overall what is changed. So using as well animal model for the disease for LAMA2-CMDs, which is this DY3K. At 4 weeks old, there was this authors performed a quantitative proteomic analysis to try to understand what was the difference in terms of the muscles between a wild type mouse and a LAMA2-CMD mouse. And you can see that there are pathways that were already established and that come up as apoptosis here in black, which represents here only a narrow slice of this pie chart. But you have here in blue, light blue and orange, two groups, two sets of proteins that in the end can be merged into the same one that play a very important role. And these are either, so in orange, generation of precursors, metabolites and energy, and in blue, metabolic process. So everything points to a very strong impairment of the metabolic program. The authors followed up this study to try to understand what was happening. And this time, instead of using mice, they also used cells, human cells that were isolated either from patients with LAMA2-CMD or from patients with Lake syndrome. And Lake syndrome is a neurometabolic disorder with a strong mitochondrial dysfunction. And here mitochondria is important because the mitochondria is the center stage for the metabolism. And that's why they try to look into mitochondrial function in these cells. And they use a machine called Seahorse, which allows, amongst other things, to look at mitochondrial respiration. And by measuring oxygen consumption rate, they can understand what is the ability of the cells to respond and to produce ATP. So there is a first step on this process where you use oligomycin to inhibit ATP synthesis. So you reduce to the minimum the oxygen consumption rate. And then you provide the cells FCCP, which is an uncoupler of mitochondrial oxidative phosphorylation. And this allows then to push to the maximum in terms of respiration. And when the authors did that, what they see is that while in the control sample, the respiration reaches over 15,000 of oxygen consumption rate, LAMA2-CMD was much closer to what they observed for the late syndrome isolated cells, which we already knew that were impaired in terms of mitochondrial function. So this was the first proof showing that mitochondrial dysfunction was important and plays a role in cells isolated from LAMA2-CMD patients. In another study, in this case, what the authors did, they used muscle biopsy of patients with LAMA2-CMD or MDC1A, which is the same disease. And in this case, they also did a proteomic analysis and tried to understand what was different in terms of these biopsies compared to normal individuals. And when they did this analysis, also what they got in terms of pathways, so they are listed here, and they do see a very strong remodeling of extracellular matrix, which if you remember what I told you about fibrosis, that fibrosis promotes this remodeling of extracellular matrix and increase of extracellular matrix. So the authors confirmed that as well with the study, but they do see a strong impairment in terms of metabolism. So they see impairment in fatty acid metabolism, glycolysis, lipid metabolism, protein metabolism, and also the TCA and respiratory electron transport chain. Okay, so everything indicating also a very strong compromise in terms of metabolism. But the studies, the way they were designed, the question still remains. So, but when does it start? And very important work from our lab by a former PhD student, Andrea Nunes, was actually designed to tackle this question. And what she found is that actually the disease onset in a mouse model of the disease called DYW, it starts between embryonic day 17.5 and 18.5. So while in 17.5, there is not much different, when we look at 18.5, there is a very big difference between what is a wild-type muscle and a DYW muscle. So if we look here into the scheme, while in a wild type context, what we usually see is that we have muscle stem cells, which provide all the precursors for the muscles. They have either maintained themselves, or then they enter into a differentiation program to allow to form then from the muscle stem cells, myoblasts, and then at the very end, myofibers that constitute the muscle. In the case of the DYW mouse muscle, we don't see that. So there is a reduction in terms of the muscle stem cell pool, and so the number of, and also the number of myoblasts, and there's a reduction in terms of muscle growth. So something is happening at this stage that does not allow the muscle to grow properly, and that may be affecting as well muscle stem cells in particularly. Another important finding of this study was to try to find which mechanism is altered at this very beginning. And when she looked into the different pathways that may be involved, so she looked at the Wnt pathway, notch, JAK-STAT, and myostatin pathways, which are all very important in terms of muscle development. She realized that JAK-STAT was altered. So there was an overactivation. So we see, for example, here in panel C, an increase in some of the targets of JAK-STAT pathway, and a very strong increase in terms of phosphorylation of STAT3, suggesting that this pathway was overactivated. And this pathway, as I said, it's part of muscle development, but is also known to act in different diseases. In terms of muscle contraction and muscle signaling, and has been linked as well to regeneration and repair. So it's a pathway that is known to play a very important role in terms of the muscle. But so this was one question, but it opens many questions, and we still have many questions in mind. So whether what is affected, is it muscle stem cells or the muscle fibers? What is actually the role of this overactivation of JAK-STAT in LAMA2-CMD? And do we have a critical role for sulfate mechanisms like apoptosis or senescence in the very beginning? Is metabolism altered, which we saw that is altered after birth? Is it altered at the moment of the onset? And what about the proliferation? Is it proliferation or differentiation? What is impaired in this context? So to try to analyze this, what we currently are doing, so with the great help of Inés, a former master student in the lab, and she helped to collect and isolate muscle stem cells and myoblasts. And these were sent for RNA-seq analysis. And this is what we are currently doing. So we have the data from the RNA-seq analysis, and we are trying now to integrate this data and to try to understand which pathways are affected. So we are doing that for muscle stem cells. And then we want to compare as well what is happening in terms of the whole muscle, understand what is the component that comes from the muscle stem cells and the component that comes from the myofibers. So in terms of conclusions, what I told you about today is that I explained what is LAMA2-CMD, and the key feature here is that it's a currently incurable disease. And we know already that chronic inflammation and fibrosis are key mechanisms activated in LAMA2-CMD, but they generally occur as secondary events. And I also told you about other studies that show the importance of apoptosis, impaired metabolism, and alterations in the expression of extracellular matrix proteins. And that our lab has shown, using a mouse model of LAMA2-CMD, that disease onset occurs in utero and involves this JAK-STAT overactivation. So now what we really want to focus is which mechanisms are altered at disease onset, and have the great picture in order to be able in the future to target it at this point and try to have new avenues of treatment and actually try to design a potential cure for the disease. And how are we planning to do this? So as Solveig introduced me, so I have a background in disease mechanisms in mouse models of the disease. and we are trying to merge all our knowledges together. So Solveig has a very, very strong knowledge on skeletal muscle development and cell exocellular matrix interaction. We count with Gabriela with all her knowledge in stem cell biology, with Rita Leão with a great help with molecular biology. We collaborate very closely with the imaging and screening facilities provided by Luís Marques and Hugo Botelho. And also in terms of data analysis, we count with the incredible collaboration of Margarida Gama Carvalho and Antonio Cordero. And we are, of course, always reaching out for other collaborations that maybe help. And we have already contacted some people to engage and promote further and to be able to succeed in our mission to unravel these mechanisms. So last but not least, I want to thank everybody. I want first to thank Solveig, which took me into her lab. It's a great pleasure to be here for a year now. And I want to thank as well everybody that has been involved in the work, either past members as Andrea Nunes, which I presented the work, and also Inês Antunes, which was very important for the RNA-seq analysis, together with Rita Leão, and also the current master students, Barbara and Pedro, and also Ricardo, which also was a master student in the lab and also contributed to some of the knowledge that we are currently addressing. Okay. And thank you all, and I'm happy now to take all the questions.
Thank you, Rita. So it's amazing work here and lots of lines of research ahead and future collaborations, as you mentioned. So we now have one question here at the YouTube channel by one of your collaborators, Gabriela Rodrigues, researcher Gabriela Rodrigues. So she starts by thanking you for the talk, and she says the results with the apoptosis knockout mouse are impressive. Do this mouse model develop cancers? Thank you.
The backs or the, because I think so and the authors, maybe that's also why they did not really go further, so they focus only in this 120-day studies, and I'm not sure what happens past that point, but yes, it's often common that mutating apoptosis pathway potentiates the development of cancer. So I'm not very sure about what is the percentage in terms of the BAX mouse alone. But for example, for p53, which is an upstream protein of BAX, the percentage is very, very high in terms of percentage of cancers that the mice develop. And usually, well, in this case, I would not expect that crossing it with a LAMA2 knockout would revert those anomalies.
My mic was off.
Was off, yeah.
And in the meantime, Gabriela has confirmed that yes, it was the backs that she was referring to. In the meantime, while we don't have more questions in the YouTube channel, and I invite, if you have, those that are watching us, if you have more questions, please feel free to leave them on the live chat. I asked Professor Christina and Professor Solveig if you have questions also on this talk, if you want also to leave them to ask questions.
Yeah, I have a question about the. So you said that the therapy that is most often used for these patients after when they're children and adolescents is anti-inflammation and anti-fibrotic. How successful are those? I'm not quite sure how successful those are.
I think those are mainly aimed at making the difficult life of the patients better, right? So they will alleviate some of the symptoms that they have. They will not revert muscle weakness, but at least they will allow to have a bit more life quality. And they often, they do as well as complementary treatments, for example, physiotherapy is actually as well very, very important for these patients, combined with these types of treatment. So it does not revert, but it slows down as well the development of the conditions.
Yeah, and maybe allows the repair to have space for repair.
For repair, yes, yes. So instead of having all the fibrosis, you reduce the level of fibrosis that you have normally in these patients.
Thank you.
Thank you. Thank you. I don't know if you have more questions here. While we wait for questions here at a live chat, I don't know if you have any comments, either Professor Cristina Maguas or Professor Solveig. We have another question here. While we wait, we have from Gabriela Rodrigues. She asks, do you know of any therapy that can be used in utero in humans, even if not related with dystrophies?
For metabolic disorders, this is common. For example, you give certain supplements to the mother. Then I'm not sure, or for example, in terms of other like infections, no, mothers can take antiviral drugs as well to prevent the child to be born with certain infections. So some are already done. So it's possible to treat mothers for certain diseases. Whether this will be we will be able to apply in this context, I don't know. Also, it depends on the mechanisms that will be identified as being important. So that's why one of the strategies that is important to take when we look at this is also to try to then look at already established drugs that can be used in humans that could have a potential application in the future.
So I have exactly a question to you, Rita, in this direction. Is it that you see in the near future, as an output of this work, a close collaboration with the pharmaceutical company in one way, and another way, if the how do you say, the clinic experiments, if you are planning to, or if you have any collaboration, close collaboration for clinic tests, let's say?
So right now, we don't have either one or another. I think we are still early on, but the way we are designing the experiments will be now in a first stage. So we will focus on the mouse model of the disease and try to identify compounds that may revert the phenotype. And then I think will be time to try to approach into a more translational study. And it will be interesting to have collaborations, and also I think at some point, and even now, earlier steps to contact to have human biopsies, because one thing is what we test in mouse, the other thing is what happens in humans. And then this is something that we already discussed, whether to also even buy some cells that are already available, human cells from patients with LAMA2-CMD.
Okay, thank you. And congratulations, Rita.
Thank you.
Once again.
Thank you, Rita, and thank you, everyone. I'm checking. For now, we don't have more questions. So if we don't have more comments or questions for now, I suggest that we wrap up for now. But the talk will be here available in our YouTube channel for people that want to check in and watch it afterwards. So I would like to thank again Rita and Professor Cristina Magos and Professor Solveig, and also Mariana that is here on the backstage, ensuring that everything is going well. So, and thank you everyone that is watching here and following the talk. So thank you, and pay attention that we will announce the next Encontros Scientia, the Scientia meeting soon. So thank you and stay tuned.